Good afternoon. In this video, we are going to pursue our discussion of the junctional complex that we started at the end of the last video on the cell. And we will start by discussing the structure of the tight junctions or the zona occludens. So in this slide, we are going to focus mainly on the zona occludens or the tight junctions. This is an external picture of the zona occludens or the tight junctions. And in this picture or diagram, we want to show you that the tight junctions or the zona occludens look like a belt around the cell. So these blue proteins connect the cell membrane of cell A and the cell membrane of cell B together, meaning that they connect the cell membranes of adjacent cells together. And we can see that the tight junctions are groups of proteins that are called the tight junction proteins that have a specific pattern on the cell membrane and that pass across the cell membrane, as we're going to see on the diagram below. So if we look here at the tight junctions, we can see that the tight junctions consist of intracellular proteins, meaning that they are proteins that lie inside each cell, inside the cytosol of each cell. And these proteins form what we call the ZO complex or the zona occludens complex. So these blue molecules are part of the tight junctions or the zona occludens, and they are called the ZO complex, and this is the acronym for zona occludens complex. But there is a part of the tight junction that is transmembranous, meaning that it passes across the cell membrane, and also extracellular. So these red proteins pass from the inside of the cell across the cell membrane and to the outside of the cell or extracellularly to join similar proteins from the other side. And these proteins are the clodins and the occludens. They are of two types. And then the ZO complex proteins are connected to very thin filaments, which we call the F-actin filaments. So you can see that the actin filaments play a role in the formation of the structure of the tight junction or zona occludens. And you can also see that the tight junctions or the zona occludens are the most superior, meaning that they are towards the lumen. And then lower down, we have the adherence junction that will be discussed on the second slide or on the next slide. And basically, the adherence junctions have the same structure, meaning that there are actin filaments to which E-cadherins adapter proteins are connected, the orange ones here, and the adapter proteins in this cell and in the other cell are considered intracellular proteins because they lie totally inside the cell. And then we have transmembrane proteins that pass across. When we say a transmembrane protein, we mean a protein that spans the whole thickness of the cell membrane and that might also emerge extracellularly to join another extra or transmembrane protein. Both are called E-cadherins, and the E-cadherins are connected together via calcium bridges that are very sensitive to calcium ions. And then lowermost, we will find the desmosomes. And the desmosomes are formed again of filaments, but these intracellular filaments are a bit thicker than the actin filaments that are present in the tight junctions and in the adherent junctions. They are intermediate filaments, but we still have the same basic structure, meaning that we have adapter intracellular proteins, and then we have the transmembrane and extracellular proteins, which are the desmoglians and the desmocollins. So just like the clodins and the occludins, and just like the ecadherins, the desmoglians and the desmocollins have a transmembrane part and an extracellular part. This is basically the structure of the three types of junctions. And now we're going to read the slides in full detail and see the specific function of each one of these junctions, and most importantly, where those specific fibers are present in the human body. So the first type of junctions, which is the tight junction or the zona occludens, they act like diffusion barriers, meaning that they prevent diffusion and they control or limit or block even sometimes the movement of molecules or ions across the cell membranes. And therefore, when they are present, they prevent the leakage of those ions and molecules into the organ or out of the organ. This is a kind of functional occlusion, but there is another physical occlusion, which means that they hold the cells tightly together like a zip. Why are we saying like a zip? Because a zip is something that is tight and that holds the two sides of a structure together in a very tight manner. And if, as we've just said, if you look at them from the outside, you will find that they look like belts around the epithelial cells. And usually, as we said, epithelial cells are cells that cover a surface. But when we talk about cells that line organs, we would rather say endothelial cells. And as we have seen in the three types of junctions, the tight junctions not only span the membrane, but extend extracellularly. And we have the clodins and occludins that join them together. And we have intracellular proteins called the zona occludens complex that are attached to the F-actin filaments. Where are the tight junctions present? They're present in the epithelium of the small intestines, the urinary bladder, the blood-brain barrier, which is the junction between the brain cells and the endothelial cells of the blood vessels. If we draw a very simple diagram of this, we would draw, for example, brain cells like this, and these brain cells have adjacent endothelial cells of blood vessels, and here is the other side of the blood vessel, or the rest of the wall of the blood vessel, and inside we have blood cells, and inside the blood we have, inside the plasma of the blood, we have substances that are dissolved. So in order to control the passage of substances from the blood to the brain, then we are not going to prevent all substances from passing, but there should be some kind of control. So this is what we call the blood-brain barrier. We can also see the tight junctions in other type of epithelia, like in the stomach and in the lungs. And then we have the zonular adherents, as we said, or the adherents junctions, and they are, again, protein complexes. We are reading the slides in order to learn how to formulate the words while answering the questions or while describing structures. So why do we say that they are protein complexes? by the way, just like the previous ones, or the zona occludens junctions were protein complexes. They are considered protein complexes because they are made of several types of proteins. And they are usually more basal than the tight junctions. And when we say basal, here we mean that they are lower in position than the tight junctions. And although the basic structure of the adherence junctions and the tight junctions are the same, yet these ones are stronger than the tight junctions, meaning that they are tighter than the tight junctions. And they are essentially specialized in like antagonizing the effects, the shearing effects of stretching, for example, or rubbing. Stretching is a good example because when you try to stretch out a tissue, you're trying to put the cells of that tissue further apart, and therefore you're trying to break down the physical connections between these cells. So when you have these adherence junctions in between cell A and cell B, so if you're trying to stretch out like this and trying to space them out, the presence of that adherence junction will minimize that effect. We have explained their structure, and we have explained that the transmembrane and extracellular, we said that the E-cadherents, the ones that appear yellow or orange here, are the E-cadherents consist of two parts, and that these two parts require calcium ions to attach to each other. So the E-cadherents are considered calcium-dependent proteins. Why? This is because in order for them to attach, they require calcium ions to bind with specific calcium receptors on them. And then we have the cytosolic side of that complex. And the cytosolic side is nothing but the adapter proteins that we have explained here and that look orange here. Those adapter proteins are the cytosolic component, and they are again attached to actin filaments. They are again present in all types of tissues that undergo stretch, and you have the list of examples, and you might be amazed to find similar examples on both slides, but this is normal because a cell will have tight junctions and adherence junctions. Maybe one type will be more abundant than the other. So it's not astonishing that we find both examples in the same cell. Sometimes in the uterus in particular, you know that the zygote divides if there is a fertilized ovum, the fertilized ovum is called a zygote, and then that zygote divides in order to form a solid mass of cells called a morula, and then the morula will develop into a blastocyst, and the blastocyst is the embryonic stage that will implant in the uterus. So in order for the blastocyst, of course, this is a section in the uterus, and these are the fallopian tubes, and the fertilized egg comes from here, or the zygote comes from here, and then we have the thickened walls of the uterus by the effect of hormones. So this is the uterine muscular wall, or a section in the uterine muscular wall. And here we have the cavity where the blastocyst will reach the uterine cavity and then will implant itself in the uterus. So in order for the uterine cells, the uterine smooth muscle cells, to allow this blastocyst to be implanted in the uterus, those adherence junction complex molecules will disassemble, will disintegrate in order to allow the blastocyst to penetrate, and then they will reform themselves once again. The third group are the desmosomes, and the desmosomes are the strongest. So you might have noticed that the lowermost ones, which are the desmosomes, are the strongest ones. They are the strongest cell-to-cell adhesions, and they are there in particular when we have a relatively wide space between the cell. So when do we mostly need those desmosomes that are the strongest ones? We mostly need them when the extracellular space in between the two cells is relatively wide. And they again consist of adapter proteins that are divided into two hemidiscs, and we can see this on the first diagram. So these are the blue ones, are the adapter proteins, and they look like two halves of a disc, and they are connected to intermediate filaments on the cytosolic side, and on the extracellular side, they are connected by desmoglians and desmocolons. So the adapter membrane proteins, or the desmoglians and the desmocolons, which we might call collectively desmoplakin, are attached on the cytosolic side towards the cytosol to intermediate filaments. But as we saw on the diagram, on the extracellular side, they are attached to cadherins. The cadherins in the middle here, these desmoglians and desmocolons, or desmoplakins, again belong to the group of proteins called the cadherins. And these cadherins, again, are two halves that are calcium dependent. They again resist shearing forces and tensile stretch. Then there is similarity not only in structure between the three types of tight junctions, but there is also a similarity in function, meaning that they resist stretch, they resist tension, they resist friction, and they prevent separation of the cells. So all types of tight junctions are present whenever we have mechanical stress. So the general name you can give to high tension, to high shearing forces, rubbing forces, all this lies under the broad title of mechanical stress. Again, they are found mainly in cardiac muscles, in the skin, in muscular tissue, where we have strong mechanical stress. Of course, in the neck of the uterus, it's very logical to find desmosomes, because during childbirth, we all know that the cervix, which is the lowermost part of the uterus, will be stretched to the maximum in order to allow the head of the baby to be evacuated from the uterus of the mother. In the cardiac muscles, when we were studying the structure of the cardiac muscles, we knew that the cardiac muscles are branched and interconnected like this. They are cylindrical but branched, and they were interconnected by intercalated discs. These intercalated discs contain desmosomes, the ones you see here, but they also contain the gap junctions that we're going to see on the next slide. So desmosomes can be present in cardiac muscles, in muscular tissue, in the skin, and in other areas of the body. But muscular tissue in general, including the cardiac muscles and the skin. Of course, when we say here muscular tissue, we mean muscular tissue other than the cardiac, because we have put the cardiac muscle as a separate entity, but they're all muscles. But the cardiac muscles are characterized by the presence of intercalated discs, and the intercalated discs are characterized by the presence not only of desmosomes, but of gap junctions. The gap junctions are, or the nexus, are protein channels, actually, and every one of these cylindrical protein channels is divided into two. It's like two hemicylinders. So they are specialized protein channels. They're only present in between animal cells. And by the way, all the previous junctions are present only in animal cells. The main function is a very rapid and direct connection between the cytoplasm of two cells. So here, the gap junctions are different from the first three types of junctions. And the gap junctions are present in particular inside the cardiac muscles. And every half cylinder is called a connexon. So the connexon is a half cylinder or half a gap junction. And here they are not there to regulate the passage of ions or to prevent the passage of ions. On the contrary, they are here to facilitate the transfer of ions between the cardiac muscles. And as you can see, each connexon passes across the intercellular space. So it passes across the cell membrane first and then across the space between the two cells to reach the other cell. And they allow the flow of ions for ensuring rapid, simultaneous contraction of the cardiac muscles. And as we've studied in human biology, this is very important. It's very important that all the cardiac muscles, upon receiving a nerve impulse, it's very important that they contract as one unit at the same time, simultaneously, in order for a more efficient contraction to occur and more efficient pumping of blood to occur. So they act as a syncytium, as we've said before. And when we say that they act as a syncytium, we mean that they act as one anatomical unit. And this ensures efficiency of the cardiac muscle. In plant cells, there is a structure that is more or less similar to the gap junctions, and that is nothing but a protoplasmic connection between the two cells. So you can find a place in between two plant cells where the cell wall does not exist and where the two protoplasms are connected. And through these protoplasmic connections, they are protoplasmic connections. They're not, it's more accurate to say protoplasmic than cytoplasmic, because they contain not only cytoplasm, but also some organelles like mitochondria and chloroplast. These protoplasmic connections allow the rapid flow of osmosis between the two cells, not only the rapid flow of water by osmosis between the two cells, but also the flow of ions. So analogous to the gap junctions in animal cells, we have the plasmodesmata that join the plant cells. So the cell wall, as it's written on the slide, is interrupted by narrow pores. Actually, they are not pores, but they are spaces in the cell wall, or spaces between the cells, between the adjacent cells where the cell wall is not present. And they are fine strands of protoplasm, actually, not of cytoplasm, that are called the plasmodesmata, and that ensure rapid transfer of water and ions between the plant cells. Just like we have talked about physical connection between the cells in the form of the tight junctions, we are going on the next slide to focus on a very important concept in AP biology, which is the concept of cell communication. Cell communication. And here we have already tackled one aspect of cell communication, which is the cell-to-cell attachment or the junctional complex, but this is a physical, more or less physical connection, meaning that out of the 4 examples in the junctional complex, the tight junctions, the adherent junctions, the desmosomes, and the gap junctions, 3 of them were essentially physical connections between cells that are adjacent. Only the gap junction is a physical and a functional connection in the way that the gap junction is not only here to connect the cells together, but also to allow passage of ions between them. But they are essentially physical. When we come to the second example of cell-to-cell communication, we have communication of cells that might be far from each other. So when we talk about cell-to-cell recognition, for example, the cells that recognize each other might be adjacent to each other, but they might be spaced out. So if 2 cells are spaced out and they recognize each other, then this is no more physical connection, but rather physiological connection or functional connection. One of the most important structures that helps cell-to-cell recognition is the glycocalyx. And as we said before, the glycocalyx consists of either glycoproteins or glycolipids. Here, for example, you can see in this example, in this part of the diagram to the left, you can see glycoproteins. They extend outside the cell membrane, and they consist of these purplish integral proteins, meaning that they span out the thickness of the cell membrane, to which sugar molecules are added. But the glycocalyx also consists of glycolipids, not only of glycoproteins. So this molecule that consists this one is again a glycoprotein. Here, the lipid refers to the lipid bilayer or the phospholipid bilayer. So we can say that all the molecules that are present here are glycoproteins because they consist of a protein moiety and a carbohydrate moiety. What are the major functions of the glycocalyx? The glycocalyx is essentially present in multicellular organisms. To start with, it's essentially present in multicellular organism, and therefore it is essentially present in eukaryotic organisms, not in prokaryotic organisms. And every individual has its own chemical structure of its glycocalyx, except for identical twins. So no 2 individuals on Earth can have the same glycocalyx or exactly from the chemical point of view, exactly similar glycocalyx, except those individuals that are identical twins. Actually, it consists of polymers, meaning that they are large molecules. And these polymers are extracellular, meaning that it's true that they start in the cell membrane and they span out the thickness of the cell membrane, but they have to extend outside the cell in order to distinguish the cell and in order to act as a label for the cell. And these polymers consist essentially of carbohydrates, which are oligosaccharides. And when we say oligo means few, means from 3 to 10. So they are short chains of monosaccharides attached to each other. And these oligosaccharides, the green ones you see here, are produced by the cell itself. And they are attached either to a protein to form a glycoprotein or to a lipid in order to form a glycolipid. So they are attached to proteins or lipids to form glycoproteins or glycolipids respectively. But if they are attached to proteins, as you see, they are attached to integral proteins, and this means integral means that they span out the thickness of the cell membrane. And they are very characteristic. I'm going upward like this. They're very characteristic because they form like a fuzzy coat outside the cell membrane of all animal cells. So we have seen that they are essentially present in animal cells and also in eukaryotic multicellular animal cells. In plant cells, we have the cell wall, but we do not have this specific structure of glycocalyx. On this slide, we can see three important functions of the glycocalyx. The first important function of the glycocalyx in general, whether we are talking about the glycoproteins or the glycolipids, that they aid in like distinguishing cell type. So they aid in cell type distinction. So although you have a basic structure for every human being that is different from all other human beings, specific protein structure, yet every cell in this particular human being will have a different glycocalyx. So since they aid in cell type distinction, they will also help in cell type recognition. Since they characterize the cell, therefore they will help recognize the cell and the type of that cell. And we cannot communicate with each other without recognizing each other. So every cell can recognize neighboring cells or even distant cells through the glycocalyx, and therefore cells can communicate through the glycocalyx. And we will see how the glycocalyx will be used not only in distinction of the cell and therefore recognition of the cell, but also in cell-to-cell communication. Another important fourth function that is here written number 8 because it comes after the description of the glycocalyx. So it is the eighth bullet on the eighth bullet point on the slides, but it is the fourth function that just like the glycocalyx helps identification of cells, of the cell types, it also helps in differentiation or distinguishing healthy cells from unhealthy cells, or our own cells from foreign cells. So the glycocalyx helps differentiate, helps the immune system actually differentiate between healthy cells and diseased cells, or between our healthy cells and invading organisms like viruses and bacteria, or between our own healthy cells and transplanted tissues that might be non-self tissues. So it helps differentiation between non-self and self, and it helps differentiation between healthy cells and diseased cells. But the glycocalyx has a fifth function, and that function is that it includes cell adhesion molecules, and therefore it helps the cells adhere to each other. And we have seen the CAM molecules before. The CAM molecules, which are the cell adhesion molecules. So the glycocalyx helps cells adhere to each other. A sixth function of the glycocalyx is to guide the movement of cells during embryonic development. Because as you know, we start as a solid mass of cells, but later on, if you study embryology in detail, which is a very interesting but challenging subject or topic in medicine, you will see that these embryonic cells will be moving in different directions in order to make cavities, in order to make layers you start by a solid mass of cells, but you end up by being an organism with cavities inside, like the intestinal cavities, for example, with different layers, a layer that will form the muscles, a layer that will form the skin, a layer that will form the skin and muscles, the bones, etc. So this glycocalyx will help cells of the same type, for example, recognize each other and be differentiated or differentiate themselves from cells that do not have the same glycocalyx. The glycocalyx is also important for a very important structure that we will be studying, or a very important phenomenon, sorry, which is contact inhibition. Meaning that when the cells get in contact with each other and feel that they are too crowded, and we will see how they feel that they are too crowded, they stop dividing. So this is what we call contact inhibition. Contact inhibition is a function of normal cells only, of healthy cells. And this explains why cancer cells, although cancer cells divide very rapidly and can therefore very soon get a very crowded mass of cells, yet they continue cell division because they lose this important property of healthy cells, which is contact inhibition. So cancer cells continue dividing rapidly, forming huge masses of cells and not stopping. They are non-stop cells. They are immortal cells. And they have the ability not only to continue division despite being very crowded. So here we can say, we can continue the sentence and say, despite being very crowded, despite being very crowded. And then they can also, because of this contact inhibition, because of this loss of the function of contact inhibition, they can penetrate the surrounding cell membranes. And they also have another characteristic that is a bit unrelated, which is they can grow without the presence of a basement membrane. Because as you know, all endothelial cells, for example, and epithelial cells grow and divide on top of a supporting basement membrane. And if they don't find this basement membrane, then they will stop dividing. But cancer cells no, and therefore they can, for example, invade blood vessels, circulate in blood, or invade lymphatic vessels and circulate in lymph without having a support from a cell membrane. One good example of glycoproteins that characterize human cells are the glycoproteins that are present on the surface of the blood cells and that determine the blood group of an individual. So if you have glycoprotein A on the surface of your red cell, then your blood will be of group A. If you have glycoprotein B, then your blood will be of group B. If you have both glycoproteins A and B, then your blood group will be AB. And if you have no glycoproteins at all on your cell membrane, then your blood group will be O. But similarly, red blood corpuscles, but at the same time, red blood corpuscles are surrounded by plasma. And in the plasma, there are antibody molecules against the glycoproteins on the surface of red blood cells. But needless to say that the antibodies in the plasma should not be antibodies against your own glycoproteins. They are antibodies against other glycoproteins, and this is helpful to prevent mixing up of different types of blood or intrusion of foreign types of blood inside your blood. So, and they help, of course, characterize the individual and characterize the identity or part of the identity of the individual. So when you are of group A, then you have glycoproteins A. These glycoproteins act like an antigenic molecule, meaning that an antigenic molecule, as you know, is a molecule that can stimulate the immune response and stimulate the formation of an antibody. So you cannot have in your plasma anti-A, because in such a case, anti-A will react against A, but they contain anti-B, and therefore you cannot transfuse blood B to anti-B. Similarly, in group B, you have anti-A. And in group AB, that is very peculiar, having both types of antigens on the red blood cell, we have none of these antibodies in plasma. And in group O, we have both, and we can have them safely because none of the glycoproteins is present on the red blood corpuscle. And here on the last row, you have the antigens that we have just talked about. Now, those glycocalyx molecules help immune functions because, as you see here, we have molecules of glycolipids on top of the phospholipid membrane, and you have glycoproteins on top of the phospholipid membrane. And these beautifully colored molecules are the exposed carbohydrate chains. Why do we say exposed? Because the proteins, though they are integral and spanning out the thickness of the cell membrane, yet they barely appear above the surface of the cell membrane. But when we say that the glycocalyx is exposed, we mean the carbohydrate portion of it. And here you can see that you have the virus that can bind to the carbohydrate moiety of the glycocalyx and the bacterium that can bind. Why do they bind? Because they have what we call lectins. What is a lectin? A lectin is a protein that can bind to a carbohydrate. A protein that can bind to a carbohydrate. So these are like receptors for the polysaccharides on top of the glycoproteins. And therefore, every cell, in order for the virus to be able to attack a particular target cell, that target cell should have the specific polysaccharide that binds with the lectin. And similarly for the bacterium. And it's interesting to know that on top of course, we're talking here about immune cells. On top of the immune cells, we can find modification of the glycocalyx to like adopt and react with new types of antigens that weren't present before. And that's why new viruses, new emergent viruses like COVID-19, for example, could attack cells and find receptors on the cells. And our immune cells could attack them because they modify the receptors on their surface. And now we're going to talk about an interesting subject, which is the quorum sensing in bacteria. The quorum sensing is a way by which the bacteria talk to each other in order to know if they are too dense or too crowded and therefore stop dividing. Too dense or too crowded. So it is a way for bacteria to communicate with each other, but bacteria and all cells in the living body communicate through chemical words, and chemical words are nothing but chemical molecules or even sometimes ions. And this quorum sensing can allow communication even between different types of bacteria, and all bacterial species actually utilize it. So it is universal. Different bacterial species can use this quorum sensing to communicate. And actually, we can say, so we might ask you why it is universal. It is universal because it is utilized by all bacterial species. We might ask you why is it considered a regulating factor for group behavior? It's a kind of group behavior because this kind of bacterial talk through chemical molecules occurs only when the bacteria are numerous enough. And when they are numerous enough, all of them contribute to that bacterial talk. And that bacterial talk allows, or this way of talking through chemical signaling molecules allows them to like detect or identify their population density, to know how dense they are, to know how numerous they are, not only to know how numerous they are, but to know whether they are numerous enough to carry out a certain function. Because as you will understand later on, there will be a certain limit or threshold after which, a threshold of density or number of bacteria, after which the bacteria as a group will be capable of carrying out a certain function. And when bacteria know about their density, when they know how dense they are, how numerous they are, they start controlling gene expression, meaning that they might upgrade, meaning increase, or downgrade, meaning decrease, the production of a certain protein by a gene, because you know that a gene is a part of DNA, and that DNA will be transcribed into messenger RNA, and that messenger RNA will form a protein, and that protein will form a certain function. And this is the central dogma of proteins. So if bacteria upgrade, they control that DNA part or that gene, they might activate that gene and therefore increase protein function, or they might downgrade that gene or inactivate it and therefore diminish that protein function. And now we will see this in detail. So what we have said is a summary of quorum sensing or bacterial talk. For example, a very discrete example and a very famous example, which is the marine bacterium Vibrio fiskeri. The Vibrio fiskeri bacterium, whose name is written here, lives symbiotically inside a variety of squids, specifically the Hawaiian squid. It's a kind of symbiosis, and we will now identify what kind of symbiosis it is, because symbiosis is a general name for any relationship between two different individuals. So the squid will provide shelter for the bacteria and will provide a source of nutrients for the bacteria, because the bacteria live in an enclosed organ inside the squid. So the squid provides shelter and nutrition. Why? Because the bacteria are there, safe inside an organ, in a very specific organ in the squid's body. But the bacteria in return will, the bacteria will in return emit light, and that light is important for the squid. So here we have a kind of, I don't know what's happening to the pen. So it's a kind of mutualism. So we have a kind of mutualism. It's a kind of mutualism between the squid and the bacteria. The bacteria give light. The bacteria are capable of producing light. And when a living organism is capable of being fluorescent, of emitting light. We will see this phenomenon of fluorescence in the unit or the chapter on photosynthesis. We call this bioluminescence because it's a kind of fluorescence or luminescence that is produced by a living organism. And this light, of course, is important for the squid in the depth of oceans or in the depth of the water volumes where it is swimming. The quorum sensing, or the talk between the bacteria, will allow them to produce this light only when they are numerous enough. And this is the importance of quorum sensing, meaning that a single bacterium cannot take or make the decision, for example, of producing light. Because if a single bacterium produces light, then this light will be very faint, will be very weak, will not be useful to the squid, and this would be waste of energy. And as a general rule for living organisms in general, they always go through their life processes towards the minimization of the utilization of energy. So quorum sensing will allow the bacteria in the squid to produce light and exert that phenomenon of bioluminescence only when they are numerous enough so that the squid can be seen and they would be effective. And when the Vibrio fiscali population lights up the squid becomes camouflaged and can feed without being attacked by the predators. So it's actually a benefit for the squid. So how do bacteria know that their population is large enough? Every bacterium will release a signaling molecule inside their surrounding environment. This is their norm. This is their routine. So point number one will occur whether the bacteria are numerous or not. So the fact that every bacterium releases a signaling molecule into the environment will occur as a routine, whether they are numerous or not. But as the population increases, the signaling molecules around the bacteria will increase. And when they increase, they will be enough, sufficient in number, to bind with specific receptors on the surface of the bacteria. So here you have the signaling molecule, but here you have the receptor. The receptor might present on the surface of the bacteria or inside the bacterium. And by the way, all the bacteria have these receptors, meaning that the same bacterium may receive a signaling molecule of its own or from another bacteria. And those signaling molecules cannot bind with the receptors until they are numerous enough. And they will not be numerous enough until the bacteria themselves are numerous enough. So what happens after these signaling molecules bind with the receptors? They will trigger a pathway of chemical reaction, or they will trigger a cascade of chemical reaction inside the bacterium. And these chemical reactions will affect the DNA and will alter the expression of the DNA. Expression means transcription into messenger RNA and then translation into a protein. So they will, here the alteration will be turning on, in the case when they are numerous, they will turn on the genes that code for an enzyme called luciferase enzyme. And the luciferase enzyme will help the formation of luciferin protein. And when luciferin binds with oxygen, it will produce light and bioluminescence will occur. we will stop here and then we will continue on the next video. Thank you.